Well, Tony, thank you for that kind introduction and for the opportunity to deliver this talk here in Michigan. I had a lot of fun reading about Jim Neal. I never got to meet him, but it was nice to go read his papers and find out a little historical facts I did not know, some of which Tony did not mention. Some of you may not know that Jim headed the Atomic Bomb Casualty Commission, but all of his correspondence are in Houston at the Houston Academy of Medicine Library, and you can actually see that there, and his careful studies besides his paper are his correspondence as leading this commission to understand the effects of radiation on people's health. Now, of course, they could not find any evidence of germline mutations, but also he had a very difficult time with the tools he had. I was excited to read about this because I'm on the committee of the Environmental Mutagenesis Society, where we're readdressing these questions 40 and 50 years later, doing trio genomes and exomes on people that have had environmental exposures and subsequent generations. So I think that the questions he was asking were incredibly important to us in clinical and medical genetics. The tools he had were rudimentary. And we are living in a time as human geneticists when it's incredible what we have available to help patients. And let's hope we always direct our thought process to that. And I'm glad to hear from the genetic counselor who just received that award that this thought of how we move it into clinic has to be very systematically looked at. So I thought I'd tell 6 short stories. About variation, mutagenesis, the same questions Jim Neal was asking, clan genomics, rare variants that we can now see, the sense for Mendelian genomics, and finally, clinical application. So, variation. I got this artwork as a Christmas gift from my daughter in London out of the Wellcome collection in their incredible museum of science and medicine in the UK. And it's really quite incredible how this resident pictured a karyotype by just a bunch of socks. And I always have trouble pairing my socks. And in fact, I settled the problem by buying all the same color, white. So I lose a pair every once in a while, but at least I can keep my socks going a lot longer. So when we think of the human genome, if we analogize it as an encyclopedia, well, the ACGs and Ts that we look at are the written language, but how does that translate into biology, and how does variation affect phenotypes? These are all questions we're very interested in, and trying to take a family-based genomics approach is the way we've looked at it. So there's single nucleotide variants, which can change the word or the meaning of a sentence. And then there's copy number variants. Trisomy 21 is one big copy number variant. And then, of course, there's indels, and the structural variants can be quite complex mechanisms. Just to summarize, when I talk about mechanisms, we want to know the structural variant, the upstream mechanisms that lead to the rearrangement, and then how does that rearrangement alter the genome to lead to disease? Is it gene interruption, gene dosage, multiple genes? Are some genomic disorders multigenic traits? We have a lot of evidence for that now. Others, single gene dosage. And I do want to emphasize one specific form of traits that was uncovered by copy number, and that's the concept of mirror traits. And here, some of genomic disorders meet with genetics. So what do I mean about a mirror trait? Deletion, you're very tall. Duplication, you're short. Deletion, you have microcephaly. Duplication, you have macrocephaly. So this concept of mirror traits is turning out to be quite important. It could also be loss of function versus gain of function. So quickly on the mechanisms. We always have to put it in the context of a timeline. And the timeline includes not just the genetic and genomic change, but the phenotypic consequences. In a beautiful paper from 1922, I pulled out by Al Blakesley from Cold Spring Harbor, he shows that this plant datura has 12 chromosomes, and he identifies trisomy for each of the different chromosomes. He coined the term trisomy. And with each trisomy, you got a different plant phenotype. So he tied the genomic change to the actual phenotype that you saw. Then the Fly group, beautiful work done by Calvin Bridges, while he was summer vacationing at the Cold Spring Harbor lab, he would take a train from Columbia University out to Cold Spring Harbor, and on the Cold Spring Harbor archives, you can find incredible things, including his last daughter in her 90s being interviewed about what it was like to live with a scientist. Very eye-opening to me to see a daughter's perspective on these things. Then finally, in 1991, we showed evidence that a duplication could cause a human neurological phenotype, not in a child, but an adult onset glaucoma tooth disease. And just like the bar locus, where it was shown that duplication gives the slit eye, and triplication gives the ultrabar eye, we actually found homozygous duplication had much more severe neurological disease. And we searched for 15 years for the triplication and reported that in 2014. So where the extra copies go shows how they will segregate, and we'll get to that. Finally, we saw the same mechanism for micro deletions predicted the reciprocal duplication, and that is now got an eponym I did not name the syndrome, called the Pataki-Lupski syndrome. I always say, I didn't name the syndrome. Lori did. If I named the syndrome, it would have been Lupski-Pataki syndrome. No, the truth is, we called it duplication 17p11.2, p11.2, and the reviewer forced us to give it a name, which was interesting. So why were these mechanisms important? First, we could see them for the first time by multiple molecular methods, including on the upper left, dinucleotide repeats, where we saw 3 alleles rather than 2. We re-looked at our southern blots and saw dosage differences when it was heterozygous. I stared at these plots for 2 years and thought this was an artifact. Never noted that it segregated in a Mendelian way until I had all the families on the same plot and reinterpreted it after the dinucleotide repeat. By post-field, we knew it was large, but we were really excited when for the first time we saw it microscopically by fish, the new technique. And then more importantly, when we got the mechanisms, they predicted reciprocal duplication, and the duplication has 3 copies of the repeat. 1 is recombinant, and we can show by fiberfish 3 copies of the repeat and 2 of the dosage-sensitive gene. Moreover, not noted by most people, is it changes linkage analysis and changes the genetics and how your computer calculates things. We only saw as a biallelic system and don't count for the dosage. It mismaps the gene. So this is my first experience with statistical genetics not showing reality. It's statistically sound, but it's not reflecting reality, because you also have to have the right input and understand the molecular and the genetics at the given locus versus the genome. So this concept of 3 alleles at locus was generalized when we found in Bardet-Biedl syndrome that patients did not get the disease often until they had 3 mutations from 2 different BBS loci. And this was exciting to me for 2 reasons. One, the first paper where we showed it genetically, we published in the American Journal. Here, where we showed it by sequencing, we published it in Science, but every figure was just pedigrees. No molecular data, just pedigrees and deductions of triallelic inheritance. So we have to think about locus and segregation of alleles and different syndromes. Upper right, Angelman, you only have 1 allele because you imprint. Stargardt is a classic recessive trait. Biallelic variation is required. DMT has triallelic inheritance at one locus, where body beetle is digenic triallelic, where digenic biallelic is RP. So these will have different mapping properties. And at the end of the talk, I hope to link this, because now we are starting to see where rare variation combines at a locus with common variation to give you the disease trait. And that common variation, you actually get a linkage signal from. So we're trying to bring together the rare variant world and the common variant world. And it's interesting, Miriam, the genetics, because you see it, because you have to be less than haploinsufficiency. But if you're fully null, you're embryonic lethal when we build it in the mouse. So you never see it as a clinician. They're dead. Very interesting. So I was amazed by the allelic distributions in the clinical population. So if we take 17,000 patients with what looks like to the clinician Charcot-Marie-Tooth disease, and you do different panels over several years, you see a distribution, but the genetic architecture is 4 genes account for 95% or more of patients. But even more remarkable, something that even just looks like CFT, some 80% either have duplication or deletion of PMP22. So CNV is much more important than SNV, certainly at the PMP22 focus. And here we show homozygous, much more severe. So we need to think about drugs that normalize dosage. And we looked at this using different chromosomal anomalies to map the dosage gene and showing contributions to different chromosomal anomalies. And Frank and I did this by just bringing patients back into the clinic that needed to be seen for follow-up. Now, in general, we have recurrent events or non-recurrent, where each event seems to be private to that family, a rare variant. The recurrent ones have an architecture that sets up the rearrangement. What about the non-recurrent? I'm not going to go into details, just published in March, but by combining long read and short read sequencing on, I think we did about 50 patients, we could show on the left recurrent events, on the right non-recurrent events, all the way to the right, the point mutations we found. So the non-recurrents happen by a replicative mechanism, which is error prone. So we see hypermutation for point mutations, and they tend to fall in genes. So it's remarkable that these CNVs can also cause point mutations in genes a megabase away, as the replicative repair polymerase during MMVR stretches out to find the next origin. Now, with the recurrent events, you can now capture two dosage-sensitive genes, and you have a new multigenic syndrome. Two dosage-sensitive genes, it grabs it, you have a mixed phenotype. These can be very difficult for the clinician to tear apart. And the counselors will be interested. In fact, it was the patients and the counselors that started to see this mixture of CMT and PTLS, and the mothers emailed me, could we study it? And I said, give me a dozen patients, and I'm happy to study it. 1 year later, they delivered 23. These things are much more common out there than anybody realizes. They're just not being diagnosed. So finally, all the way to the right, when the Watson genome was done at Baylor, and I was chosen as the person to counsel them as part of the IRB requirement, I was struck by the allele fraction of CNVs, and as you got smaller, the frequency was tremendous. And this was 454 sequencing. So we had 250 base pair reads, and we could get through ALU. So there's a huge peak of ALU. So ALU dimorphisms is one of the most common mutational events in your genome. Now, how are we going to figure out what's happening in the gene? So, first thing we did was put them in yeast and show in the break-induced replication assay, they work better than yeast repeats. We did this with greater IRA. So it really shows that these template switches have much different requirements than homologous recombination. And that's why we can drive so much recombination with this mechanism. Then get a good student interested, and she does machine learning, but because there were 78 million pairs of ALU, we had to use a supercomputer at Rice. And she's developed this wonderful algorithm that predicts genomic instability by ALU for 12,000 genes. And it's very hopeful for us then to look at the patient data on the exome, at the ones that are very unstable. Now, how do we translate this into parents and phenotypes? Parents want to know, what is this? Why did it happen? What's the chance it's going to happen again? So Lori came up with this nice picture, which is very helpful, about the normal sentence, I like to swim in the ocean, but I do not like to swim in the pool. A deletion, I like to swim in the pool, still has one copy of the repeat. Duplication, it has one copy on the normal chromosome. Duplication has the orange duplicated, but the white is actually duplicated. What is a mere trait? At the parent support meetings, some of the SMS patients started to come to the PTLS meeting, and they asked us to take their picture. Both of these adults, it's pretty striking how one's tall and one's pretty short. We then engineered in mice the same segment. Chromosome 11 is highly synthetic to 17. We use chromosome engineering, and note the duplication. It's the same weight issues. With SMS, you get obesity, PTLS, they're always very thin. So we see each dot is 10 animals and their weight over time. So this is truly a genetically determined trait. So now let's feed the mice Big Macs and look, the PTLS mouse does not get fat no matter what you do to it, but its litter mate sibling gets fat like crazy. So here you see environment with genetics. Why is this interesting? Because at a meeting of the National Academy celebrating 150 years of Darwin, Bernie Crespi presented the latest theory on behavioral traits from the social anthropologist viewpoint. If you look at primates, the size of their brain is directly related to the size of their social unit. We as humans spend a lot of time interacting with other humans, and we have lots of behaviors related to that. There were 4 general theories put out there about autism and psychotic disorders. But Bernie Crespi was the first to put out the idea that they're just the extremes of normality, and they're the mirrorest traits of each other. So we had just shown that duplication of 1q21 gave you the opposite head size as deletion, and then the deletion was linked to schizophrenia, the duplication was linked to autism. Meirwan Chenaui, as a fellow at Baylor, also showed at the 16 P11.2 that in this case, deletion was microcephaly, duplication was, deletion was big, macrocephaly, duplication was microcephaly. And then linkage showed deletion was autism, duplication was schizophrenia. And Bernie now has many loci around the genome where loss of function gives you one, gain of function gives you the other. Now, I can't get a single psychiatrist at Baylor College of Medicine to send an array. Meanwhile, in our diagnostic lab, we diagnosed over 500 families with deletion or duplication at 16p. And beautiful work at Alex Raymond's lab shows that when you go into biobanks, the Estonian biobank, you can find many genomic disorders that the clinician never recognized. And now you have full records, and you can clearly demonstrate performance changes or cognitive traits based on these CNVs. Plant genomics. I just love going to the Galapagos. A biologist dream brought my children, but I've also now spent about 8 years going back and forth to the Faroe Islands. I didn't even know where they were, but all 126 physicians use the same computer system, as do the 2 hospitals and all the pharmacies. So this is where we've been trying to integrate genomics into practice directly. And the Faroes is the Galapagos of the human race. Halfway between Iceland and Norway, they have been isolated for hundreds of years on 26 different islands. I don't have anything to report on it yet. But this mechanism of MMBIR is turning out to be very important for evolution. Gene duplication and divergence, amplification, exon shuffle, new gene creation. So we recently reviewed all the different combinations you can get from 2 template switches rather than 1, 3 template switches. And then the bottom in the middle, what happens when you switch between homologs versus sisters? You actually get UPD, but that whole region you replicate through. When you do trip dupe and you invert the segment, you create new genes different from triplication by NAHR. And it's a very complex structure where you can see the 2 junctions make fusion genes, but the structure is quite complex and not deconvoluted by most of the sequencing programs. Now, here shows dupe, trip, dupe, followed by absence of heterozygosity because of the template switch above other SNP arrays. And you see you lose the B allele frequency. It's absolutely amazing. So when you do this, you can look at each of the genotypes and even see where you go from AABB to AAAB, and that shows a template switch without a copy number change. Amazing. This shows such an event. Now, you say, so what? This is like incredible theory. But we predicted if it happened through an imprinted locus, you would get a syndrome. And that's just come online in genome medicine. Temple syndrome by this exact mechanism. So the clinical lab really has to dig into their data beyond just what the copy number variation, but it's also the SNVs happening as part of the mutational event, or are you template switching to the homolog and going through an imprinted region? All of these things, imprinting, these are all gene dosage phenomena. As I hate to tell you, is recessive versus dominant trait. Recessive, you need 2 copies of the mutation. It's gene dosage. Dominant, just 1. Now, interestingly, we tried nanopore, and when we can get sequence reads through the whole event, we can computationally deconvolute dupe, trip, dupe, as you see on the right. All done by nanopore. Now, why did I like nanopore? Because I get the methyl groups. So I can look at my patient and I can see the methylation changes, and I can see the loss of imprinting. And it corresponds to bisulfide arrays. So there's more information I can get out of the SNVs by looking at the methyl cytosines. And I'm not going to go into it, but the extreme of this mechanism is chromothripsis. First seen in cancer genomes, but we also saw it in germline mutations as de novo events. And I point you to the papers, but I made the slide from a News and Views, which I think in 2 pages describes the discovery better than I ever could. So what is clan genomics? Rare variants, highly penetrant, are associated with Mendelizing disease traits. They have arisen recently, and evolution has not purged them from your personal genome. But you need to have them to make the variation that can start to see if you're better fit for that environment. So conceptually, it's that heat map on the right. The top right shows different disorders, chromosomal, Mendelian, genomic disorders. When we're dealing with a finite genome, the variation has to be there to cause it. So the heat map shows, look for the de novo events, or those that might be recessive alleles came from either parent, or those that by IVD may have come back on the parents in the clan for that mating. So it's very important to understand this, because this is the theoretical basis of family-based genomics and rare variant analysis. So I just pulled case reports from the literature. A family, cousins with CMD, but you go and do molecular testing, and the twin has a de novo duplication. The cousin, which actually is from a consanguineous meeting, has IVD for a recessive locus. And we started to see this more and more. And we started to see mutations in 2 CMD genes. But we can also, from the clan genomics perspective, get at the variation by trio analysis in the twin and by quad analysis in the small nuclear family with the recessive. As you can see, IVD brings back the recessive allele. So we developed the genomic sequencing in a very systematic way over a 5-year period at Baylor. And this was a plan. Richard, Eric, several of us got together. And I want to say that my genome turned out to be important because it's the first time we were looking for variation that we could link to the phenotype. The Beery twins, we wanted to find variation causing the phenotype, but we could treat it based on that molecular. Then we moved it into the clinic and had clinical sequencing and research sequencing. So in the CMG, this review just came out, but we've done 61,000 samples, okay? 22,000 families, about 50,000 exomes, and found lots of genes. And many of them are making it into the literature. I think important, 4,000 collaborators, right? Incredible. So here shows you the statistics. And by linking into GeneMatcher, you now link into the world of variants. And soon, you should be able to go from GeneMatcher to Variant Matcher and directly into our databases. 260 novel genes have been published already from the effort. Lots of gene matches all over the world. Now, the way I've done it is different philosophies. The genomicist can teach the clinicians genomics, or You take the clinician seeing the patient, you convince them to do research on their patients, and you bring them into your own lab. So when I went to Istanbul to speak in their national meeting, they put me in a fancy hotel room. They wanted me to meet with all the elders. I said, give me your 12 best clinical geneticists in Turkey at this meeting, fellows, and I'm having them for lunch in my fancy room overlooking the Bosphorus. And they came, and since 5 have come to my lab. We've done over 1,000 Turkish families. Published. This is just for the NIH type people. They keep saying, I'm not sure if they're going to renew the program. Published 570 papers. In a 6-month interval, that went from 570 to 620. I was just searching on our grant number. Finally, clinical genomics. As we saw the field move, a lot of the students don't realize we knew the structure of DNA before we knew the right number of human chromosomes. And chromosome studies were very important in the development of clinical practice, as has been structural variation. Now, but conceptually, a big change was when we went to gene panels, checking many loci at once, rather than a single locus. And we also started this with telomere fish. Rather than one fish locus, we went to all the telomeres. Then we had arrays, and we could see the whole thing. And now we have sequence, and we can see the whole thing, but most of the time we have no idea what the hell it means. That's getting better. The first 2,000 at Baylor, 25%. Paper coming out the next few weeks, you will see this number, those patients going to 38% just by reanalysis once every 6 months. Computational reanalysis. The original New England Journal paper, which was 5 years out, has gone from 24% to 48% in 5 years of reanalysis. So you have to establish the genomics with your population, your clinical population. I don't care if you do that by outsourcing or you build it yourself, but it's necessary. Now, incredibly, if we look at the autosomal recessive, I was taught they should be compound heads. We find a third are homozygous. 3% are homozygous because of UPD, not because of Mendelian segregation. So, missegregation of chromosomes that might lead to aneuploidy and then trisomy to disomy rescue happens a lot more clinically than you realize. Excellent. I thought it'd all be males. And 1/3 of the males would be de novo. Instead, we found half were males, half were females, and 74% were de novo in the females. So there's theory, and then there's reality. Clinical medicine is reality. Then you start finding the patient with the array didn't make the diagnosis. They had an exon dropout, and mom had it. But now you do an exome and you find a point mutation in the gene they got from that. So now you've gone from a recessive carrier state to a CNV over an SNV. And nice clinically is the single exons are highly quantitative by digital droplet PCR. So like you use Sanger for SNVs, we will be using ddPCR and in parents to make sure they're carriers when you see homozygous deletion. And combining the research lab with the variant information from the clinical lab, they synergistically feed on each other. So our consent, the patient or mom or dad can check a box that says if a molecular diagnosis is not made, do you want your data moved to the research lab? 99% of parents check the box. Of course they want an answer. Of course they want research to be done. They don't want to hear about HIPAA. They don't want to hear about the IRB and what they want. They want a damn answer. Let's give it to them. So we take the unsolved clinical exomes, and 75% of them we can solve by new gene discovery. And this is the paper that's about to come out. 75-page paper. They allowed us their new format, 750 words. One failure. They allowed us two. This was one of the ones. So the journals are moving towards this thing that everything is supplementary. But it does force us. What is the key point? The key point is reanalysis is important, and most of it is new gene discovery. And we can do the same thing with the CNV data from the array lab. Now integrate that with the exome data, and we have 3,000 patients that have had both. And we can see what size CNV you can get from the prediction programs from the exomes. Because let me tell you, generally they suck. And they give you so many false positives. There's no way clinical sign-out can chase them. So we demonstrated this with homozygous exonic deletions where we could use the parental carrier state to also show the genetics. And this just shows that currently at least 32% of clinical causative CNVs are not being signed out by any exome data. Also, we can start to see the different phenotypes. So if we look at cohesinopathies from a variant standpoint versus only studying those with Cornelia-de-Lange syndrome, we start to see how the different subunits of the cohesins present slightly differently, and the phenotype is not quite CDLS. Very incredible. And with the dual diagnosis, integrating that with systems biology, we can try to see when you have a blended phenotype, is it more likely to be a discrete set of phenotypic characters or overlapping phenotypes, making it more severe. And the most amazing thing is that for dual diagnosis, when it's monoallelic, autosomal dominant, X-linked in males, 44%, both mutations are de novo. So there's no family history that helps you with anything. And we can start to use dual diagnosis to separate out in the family intrafamilial variability. Some of the multiple diagnoses here was because an AOH block caught all the genes, but the AOH block might just do a subset of genes, or you have a family member who's slightly different and only has one gene mutated versus the 2 or the 3. So sometimes you should be thinking about doing exomes in multiple affected members of the family. And as you move into populations that have more consanguineous ratings, you see a shift from double de novo to AR plus AR, because the clan genomics and the identity by descent happens quicker with those pathogenic variants than the de novo mutations at the locus. So we think that clan genomics also explains a 20-year paradox in human genetics known as the union paradox, where when you did linkage mapping of founder effects for limb-girdle muscular dystrophy, you found the same founder haplotype as you would expect. But when you sequence, there was many different point mutations. So you have the founder haplotype, but mutations happen on the founder haplotype. So the founder haplotype comes back on itself, but now in one family, it's one mutation. Another family, it's another mutation. One last point. We saw that congenital scoliosis in 12% of Chinese patients was one gene, DBX6. And you had to have a null allele, which most often was 16 p11.2 deletion, where the gene maps. But if you got the deletion, you didn't always have scoliosis. And if you had scoliosis, sometimes your parent didn't have scoliosis. You only got scoliosis when you had the 16p11.2 deletion, either de novo or from a parent, and you got the GWAS haplotype from the other parent. And haploinsufficiency never gave you disease, but you make another allele, homozygous, and it's dead. The bug did that in mice. He used CRISPR to make the GWAS allele, and then CRISPR to make the knockout allele. Then mated all these mice. Only when you have the hypomorph, which the GWAS signal is an EQTL, it changes the expression a little with the null. Do you see scoliosis in the mice? And you distort the genotype ratios, and you see embryonic lethality of the double null. So in human populations, you don't have to have homozygous. You can get a hypomorph allele, and in the Chinese population, 44% have that haplotype. So the penetrance is a function of the hypomorph. Or another way to think of it is it's a recessive trait, but one allele is severe coding, the other allele is mild hypomorph, non-coding. But we need a non-coding variation, and we need to bring together the world of common variants and rare variants. So in the mouse, you reproduce what we saw in China, what we saw in Japan, what we saw in the USA, including which part of the spine is involved. And this is all the most talented orthopedic surgeon I've ever worked with, who on a fellowship came to my lab for 2 years to do this work and set up his lab in Peking, where he operates on hundreds of scoliosis patients from all over China. So they selectively are brought to him for surgery, and then we all do the molecular together. So in summary, plant genomics is an important theoretical framework. It's not just more of the same. It links evolutionary biology to real-world genetics of the clinic. I'm not trying to say rare variants are better than common variants. I'm not trying to bash GWAS. I'm just trying to get at the truth. Structural variant mutagenesis is important, and we're missing all the exon dropouts. DNVs, imprinting, UPD, compound inheritance, EQTL, biallelic, monoallelic, it's all a gene dosage phenomenon. Whether it's dosage of the wild type or dosage of the mutant allele. 1 in 20 molecular diagnosis at Baylor. Now we've done 14,000 exons. This is a dual diagnosis. Multilocus pathogenic variation is important, and we believe we can build models for the genetic architecture of disease, not from top down of disease population, but from bottom up, from genetic variation and phenotypes we observe in the hunt. The proudest moment in my life was when my first 2 graduate students organized a 30-year reunion, and the 75 postdocs that have gone through the lab and the 25 PhDs I've graduated, 97% of them showed up for the meeting and then the banquet from around the world. I love being at the meetings, seeing them thrive throughout the world, and visiting very nice places. It doesn't happen without great colleagues, students, and fellows to have a lot of fun with. Thank you for listening. I'll take questions if there's time. I'm glad we got the AV set. Were you able to broadcast?
Thank you so much, Jim. That was just a beautiful talk that touched on so many important topics. So we definitely have time for a few questions. So maybe I'll start off with a more philosophical.
With the Greenberg lecture, we put it up on YouTube.
Yeah.
And I'm actually impressed the number of people who are pulling it down.
So you have shown so beautifully over and over again that there probably is no such thing as a simple Mendelian disease. So for people interested in rare disease research, where do you see the field going in the future, and for clinicians and basic scientists?
I think the field is exploding.
Yep.
Okay? And I think it's a misnomer to think that you've got to be on the gene discovery paper to do the functional analysis. As I see it, the gene discovery is the first part. And there's tremendous amount of biology that the real scientists have to dig in and do. Right now, we're so overwhelmed with sequence data and interpretation. My focus is to just get out the discoveries. And I've closed my mouse work, and we do a little functional work, but we're trying to use genetics and the power of that to deal with the next 17,500 annotated genes that have no functional variation associated with them yet. 17,500 annotated genes have no function assigned to them.
Thanks, Jim, for a really great talk. I have a question about what is from your perspective, how do you go about trying to inhibit PMP22 to benefit patients that have a CMT1A duplication or triplication?
Well, ISIS has had a discovery project on this for a long time. They're starting to have molecules that do lower the expression. And the first thing is showing it effectively in the animal model. And that work is still ongoing. But yeah, you don't want to overdo it because you get the HNPP. But HNPP is not as bad a disease as CMT. So you have to play around and titrate things. It's going to take time, Jim. But I think it's amazing that we have the molecule now to start. So I would say for those who are new scientists, don't worry, there's going to be plenty of work to do. For those who are inclined to physician scientists and really want to have their hand in clinical work, the tools are going to be amazing. And not just from the diagnostic standpoint. I think just dealing with the stop codons, there may be ways to manipulate beyond what people have thought with premature termination. With imprinting, look at the way I published that nice paper on treating by taking away the imprint using an oligo. So I'm so excited about the future. And I just think about people like Jim Neal. They had such little tools, but what they did was amazing. And some of the same questions still have to bother us in terms of as we start to dissect gene environment interactions, which I think we're starting to do. It's going to be very important to use that in a clinical treatment sense, but also in stopping us putting things in the environment that might do harm. And I'm not just talking about nuclear, I'm talking about lots of other things.
Great, thanks.
For the current students to take on.
All right, so let's thank Jim one more time for a really wonderful seminar. So we will be having